Okay, thank you for introduction and thank you for invitation for the webinar. And I'll tell you a little bit about multiphoton microscopy and translational preclinical neuroscience. And Dr. Ettenberg already introduced some of the features of multiphoton microscopy. And this is a very specific type of microscopy, which was invented like a couple of decades ago, and it's very well suitable for preclinical and translational research. It's based on phenomena described by Maria Guppert-Meyer in last century, who showed that when two photons hit the fluorophore at one time, they cause an emission of a photon of higher energy. And in that case, fluorescence occurs only in the focal plane. And as you see here, this is a multiphoton fluorescence in one point of focus, and there is a standard single photon fluorescence where you can see the fluorescence across the whole specimen, which gives very bad noisy background and also add for phototoxicity. So this is a schematics of the multiphoton microscopy versus confocal microscopy. And in confocal microscopy, the good image resolution can be done with a change in pinhole, while in two-photon microscopy or multiphoton microscopy, you don't need it because you have fluorescence only in the focal point. And also it has a better penetration because it's the excitation occurs at infrared or near-infrared spectra, which has a deeper penetration. And I'll describe you a little bit about one of my projects where we use multiphoton microscopy for detection of cerebral microcirculation heterogeneity in traumatic brain injury and intracranial hypertension. So as you all know, brain injury has an exceptionally high energy oxygen consumption, and a high proportion of cardiac output is going to brain, and it extracts about 35% of the brain as opposed to the 25 in the rest of the body. And of course, it's all depend on microcirculation, where main oxygen and nutrient exchange occur. And so why I'm talking about intracranial pressure? So I need to give a couple concept of it. So intracranial pressure is the pressure exerted on the brain tissue inside the skull, and it's about 7 to 15 millimeters of mercury in adult at rest. And the relationship between the contents of the cranium and intracranial pressure is described by Monroe-Kindle doctrine. So what is this doctrine? This doctrine states that all intracranial components, CSF, blood, and brain parenchyma are enclosed in the bone compartment, which is constant in volume, and therefore total volume of the intracranial components remains constant. If there is any change in the volume of any of the component, for example, tumor or hemorrhage, it should be compensated by reduction in the size of on the volume of other compartments, which is in compensated states. However, if the intracranial volume of one of the component increases too much and there is no more compensation, and due to the rigid of the bone cavity, the intracranial pressure raises. And this leads to the pathology because it increases intracranial pressure. And intracranial hypertension is one of the most damaging aspects of the brain trauma, which occurs in 50% of TBI and correlates with poor outcomes. And it increase in intracerebral compartments cause ICP elevation, which reduce cerebral blood flow, and it's a vicious cycle of increase of intracranial pressure and brain pathology. And there is no evidence-based standard of care guidelines for this pathology. And another concept is a cerebral autoregulation, is a brain ability to compensate change in arterial pressure to maintain cerebral blood flow within the cranium, and it has the limits of autoregulation where vasodilatation and vasoconstriction cannot occur anymore, and after this arterial pressure parameters, there is no compensation, and cerebral blood flow decreases, which causes to brain ischemia. And how it was originally described, it was described by autoregulation curve, where arterial pressure was gradually increased, and by measuring cerebral perfusion, it was defined that critical level of cerebral perfusion pressure is 50 millimeters of mercury. However, in clinical settings, cerebral perfusion pressure, which is a difference between arterial pressure and intracranial pressure, usually decrease because of intracranial pressure increase. And it was discovered that there is a phenomenon with when you decrease cerebral perfusion pressure by intracranial pressure increase, it shows better preserved cerebral perfusion pressure limit, which was about 45 to 30 millimeters of mercury. And there is a problem. The low limit of cerebral blood flow autoregulation obtained by arterial pressure is about 50 to 60 millimeters of mercury, and when you obtain it by increase of ICP, it's about 30 to 40 millimeters of mercury. And it suggests better preserved CBF autoregulation with increased ICP, which is ridiculous because ICP is a secondary pathological complication. And this is a clinical manifestation of this phenomenon. You can see the MRI of the girl under the barbiturate coma with infarcted left hemisphere, and despite the barbiturate coma, you can see a very high flow velocity of blood flow volume in infarcted hemisphere, which should not be like that. And we hypothesize that a decrease in the lower limit of cerebral blood flow regulation with increase in ICP occurs due to a shift from capillary blood flow to high velocity non-nutritive microvascular shunt flow and high vessel diameters. And cerebral microcirculation becomes more heterogeneous. And we aim to show that this was falsely improved as measured by Doppler probes, which just do average of all vessel flow velocity in the tissue. And for that, we used multiphoton microscopy, and I would emphasize that we measured microvascular flow velocity in each capillary and NADH fluorescence to show the brain metabolism and oxygen consumption. So this is our experimental setup where we measured all physiological parameters, and using multiphoton microscopy, we measured the flow velocity, BBB permeability, and NADH as a measure of oxygen. So this is a area of our imaging, and we controlled it by laser Doppler flow to show clinical measurement. So what we did, we decreased cerebral perfusion pressure either by arterial pressure decrease or intracranial pressure increase. And we show, our Doppler flow probe shows the same phenomenon. You see that the cerebral blood flow is more preserved with ICP increase. However, at the same time, with an ADH imaging, which was done simultaneously with cerebral blood flow imaging using photon microscopy, we showed that in a group with increased ICP, the tissue hypoxia was much higher than in arterial pressure decrease group. The same thing we demonstrated with blood-brain barrier, which was disrupted more dramatically in a group with increased ICP as opposed to arterial pressure. And the same thing was the brain water content or edema, which was dramatically increased in the ICP group where we increased ICP. So what was the reason for this phenomenon? And we were able to show this with imaging of blood flow velocity in each capillary and blood volume of imaging. So we use blood flow plasma labeling by tetramethyl-rhodamine EZ cyanide dextran, and we use so-called line scan measurement, where we can measure blood flow velocity in each capillary by line scan. And here you can see the frequency histogram of a frequency of occurrence versus flow velocity in capillaries in small vessels, which has a normal distribution. And what we found is that when you increase ICP, the microvascular flow become heterogeneous. So the most capillaries become low flow velocity, but at the same time, we found the group of vessels with larger diameter that become very high flow velocity, which we call microvascular shunts that are non-nutritive and pathological, which was not observed in arterial pressure group. So we create a hypothesis how it works, and we conclude that decrease in critical cerebral perfusion pressure with increased ICP is associated with the redistribution of blood flow from stagnant capillaries to pathological macrovascular shunt, leading to tissue hypoxia, BBB dysregulation, and cerebral edema development and neuronal death. At the same time, reduced blood flow in capillaries and non-functional hyperemic blood flow of microvascular shunts are averaged by laser Doppler in clinics and seems to be normal, which is the reason for false decrease in the critical low thresholds of CPP. And this was also described later on by Leo Ostergaard, who developed a hypothesis of heterogeneity based on our discovery. Seems I'm getting out of time. Right? So but multiphoton microscopy still is not usable for clinical research, and we found two solutions how to find this, how to do it in clinics. We use induced cerebrovascular and intracranial pressure reactivity index, where we increased arterial pressure by small pulse, which was adopted from Dr. Rosenthal method, and we shown that in that case we can predict the occurrence of microvascular shunts, and we tested with animal model of traumatic brain injury, which clearly predict the microvascular blood flow heterogeneity. And also solution number two, we use high resolution MRI, we use a voxel analysis of traumatized tissue, we're also able to predict it. And I'll skip the conclusion. And I want to present a couple more examples. So in this case, we did in vivo awake imaging of mouse under transcranial alternating current stimulation through intact skull. So we use so this is an intact skull, and we treat it with a series of solutions, so it become transparent to the optical light, and you see that the arterial diameters and change in arterial diameters in response to stimulation is very different in awake versus anesthetized mice. So this is why we're moving toward imaging of awake animals. And I just was excited by Dr. Rowe's presentation, and this is our imaging of dendrites, miniature spontaneous spikes using 2-photon microscopy with argon BAPTA dye, and you can see with very high resolution the change in calcium in dendritic spines. Okay, thank you for your attention.
Thank you very much, Denis. That was lovely talk. And we have some time for questions. Any questions from the audience? Jake?
Hi there. Thank you for your talk. I was interested in one of the last slides that you showed about the differences under isoflurane anesthesia. Did you find that with different types of anesthesia that are used for mice, like, well, any other anesthetics that you've tested?
It would be very good to do that, and I didn't do it specifically in this experiment, because in this experiment we use a standard azofluorine anesthesia, which, as you know, cause some vasoparalysis. It's not complete, but it's reduce reactivity of vessels. But I also tried with urethane, which does not affect vascular dynamics, but significantly affects brain metabolism. And I also tried propofol, but this was just short trials, and I didn't do it as a whole study, which might be very interesting.
Gotcha. Thank you.
Have time for another couple of questions, maybe. I have a question. So Denis, you're imaging dendrites and you're also working on traumatic brain injury. I'm curious, have you ever looked at what happens to neurons and spines when you increase intracranial pressure? Is it well known what happens? But I would imagine you probably would disrupt some synapses.
Yeah, of course, it disrupts some synapses, and it's cause neuronal degeneration in general, and since it causes also cerebral ischemia, it affects it. But I didn't do specifically imaging of dendrites, which might be also interesting to do. This is just example from my previous work while we studied synaptic vesicles release due to stimulation.
So you increase, I mean, I would imagine you would increase spontaneous vesicle release under conditions of increased intracranial pressure.
No, it's just I just added this example. This work is under review, and we did study readily releasable and docked vesicles release using a specific dye which called FM143, which, for example, Dr. Stanton is doing pretty. It's in his fields of interest, and in addition, we did a calcium spinal mini PSP. So it's not translational work. We did it for on a normal brain slices.
Thank you very much. So any more questions? If not, let's thank him, Denis, once again.
Okay.